Okay, today, guys, we're going to talk about membrane transport. If you remember from previous lectures, the cellular membrane is going to be selectively permeable. All right, so there's two ways you can get across the membrane. The first way, or the first one we're going to talk about, is what we call passive transport. Okay, and by definition, passive transport is going to mean no energy required. Okay, so things are going to move without energy. And so the only way that things can move without energy is if they go from a state of high concentration to low concentration. All right, so if you want to think about it like stairs, okay, passive transport is going to be things moving from high, so if this is like the concentration, okay, so they're going to be able to move down the stairs very easily, from high concentration to low concentration. All right, but instead of being in stairs, we're going to look at the movement going across the plasma membrane. So remember, this is going to be a phospholipid bilayer. And I'm just kind of drawing it quickly. And so that movement is going to be across the membrane. So what we would have to have for this passive transport to occur is a high concentration of the substance on the outside and a low concentration on the inside. And if it's able to move through the membrane, we're going to call that process diffusion. Okay? Diffusion is movement across the membrane from high to low concentration. Okay? If you remember this hydrophobic, this lipid bilayer, some things that are polar are able to go through there very easily. If they're small, or excuse me, nonpolar and small things are able to go through the membrane easily. All right? But sometimes that diffusion, because of the chemical properties of the lipid membrane, is going to have to be helped. And so we call that facilitated diffusion. So it's the same thing. It's movement from high concentration to low concentration with the help of membrane proteins. So we're going to have movement from high to low with protein help. Okay? And literally inside of this membrane, we're going to have what we call these protein channels. I'm just going to draw a crude drawing. And so what that's going to allow it to do is, let's say this is a polar molecule, like water. Okay? Water is actually able to move across the membrane, but it does so through protein channels. Actually, aquaporins is the protein that they get to go through. And so they're going to be able to make their way from high concentration to low concentration through these protein channels. All right? And that is called facilitated diffusion. Both of those belong under passive transport, moving from high concentration to low concentration. Okay? One type of diffusion that's very important for you to learn, and we kind of touched on it right here, is the movement of water through the membrane. This movement of water through the membrane is called osmosis. Okay? So, movement of water across the membrane. Now remember, it is still from high concentration to low concentration. Okay, so, and if you also recall, on outside of the cells you have a water environment, an aqueous solution, and on the inside of the cell you have an aqueous solution. And so we actually get into 3 types of osmotic situations. Okay, and so I'm going to draw 3 beakers. And each of these 3 beakers is going to represent a different aqueous solution. Okay, remember that means water is the solvent. Okay, so I'm going to have cells in the aqueous solution, and obviously these are huge cells. There would fit millions of cells in a beaker of aqueous solution. And so the water, I've got to draw the water in here. Okay, so these cells are put into this solution. Now, like I said, there's 3 types. And so let's say that the average human body cell is 85% water. Okay, so in this first one, I want you to put 85% water, and that leaves 15% of that cell is going to be solute, is going to be things dissolved in the water. If only 85% of it is water, then that means 15% of it is going to be dissolved things in the water. Okay, now let's say that it's placed in a solution, so that's the inside of the cell. Let's say, and I'll change colors for you, the outside of the cell is 90% water, which then only leaves 10% for dissolved substances. Okay, when we talk about the osmotic effects, I want you to think of the net movement of water. Okay, so I'll write that right here. Net movement of water. So if I have 85% water on the inside and 90% water on the outside, the net movement of water is going to be into the cell. Okay, so the net movement of water is going to be into the cell. We call this situation where water is going into the cell a hypotonic solution. Okay, so that's your vocabulary word. Hypotonic solutions are when water goes into the cell. What are you going to see in hypotonic solutions? You are going to see the cell swell. All right, so when water is moving in, it gets bigger. All right? The next one, let's do the same thing. We've got 85% water. That means 15% solute. Let's go ahead and put it into a solution that has, say, 80, just finger erase here. Okay, let's put it into a solution that has just 80% water and 20% solute. All right, so the net movement of water, then, if I have 85% on the inside, 80% of the outside, I'm going to go from high to low, and so water is going to leave the cell. So the net movement is out of the cell. Okay, that means more water is moving out than is moving in. And we call this hypertonia or hypertonic. Okay, what is the result? What are you going to see? You're going to see the cell shrivel. All right, so in theory, if you have a severely hypotonic solution, you could get the cell to swell so much that it bursts. In a hypertonic solution, you could get the cell to shrivel so much that it's not going to be able to survive. It's not going to have enough water. It's not going to have enough enough room to do what it needs to do. Okay, the third type of situation we could have, let's say we have 85% in, 15% out. Let's put it in a situation where it's exactly the same. And this isn't a trick question, but if I were to ask you which direction do you think water would move, I need you to tell me. Okay, water would actually move in and out of the cell, all right? But the net movement of water would be 0. Okay, so even though we have movement of water, there's just as much moving in as there is moving out. Okay, we call this situation isotonic, and so you're going to see the cell be at a constant equilibrium. Okay, so with hypotonic, we're going to have water move into the cell until we reach equilibrium, okay, or worse if we are in an extreme situation. Hypertonic, it's going to leave until it reaches equilibrium. At an isotonic solution, it's going to reach, or it's going to be at equilibrium. Okay, so that is all passive transport. No energy required. Okay, there are two types of transport that we call active. So I'm going to write those over here in the corner. Okay, I want you to remember that active transport is the opposite of passive, and so that means energy is required. And like I said before, there are two types. Okay, the first type is going to be on the molecular level. Okay, so we're going to be moving molecules. Instead of going from high concentration to low concentration, we're actually going to have to move molecules from a low concentration into a higher concentration. So the staircase is going to be opposite. So instead of moving down like we do here in passive, we're going to have to move up. All right, so if we're on the outside of the membrane, so once again, let's get that sketched real quick. I'll just draw 5 little phospholipids here. Let's make the tails. What active transport means is that we're going to want to move. There's already, let's say this is a muscle cell. This is one of the areas where we have active transport. In muscle cells, we have a lot of sodium on the outside of this membrane, okay? And we have very little sodium on the inside of the membrane, all right? But we're going to use pumps, okay? And so on a molecular level, we're going to use pumps. They're called ion pumps to move molecules from low concentration to a high concentration. This basically, physiologically, is going to cause a chain reaction. If I flood an area with these ions, then at some point they're going to have to come back through. And so this potential energy, right, if we're putting all this energy up there, at some point we're going to get some kinetic energy. All right, so on a molecular level, we use pumps. There's also a bulk level transportation that requires energy. Okay, so we're going to call that bulk transportation. And these two processes are endo and exocytosis. If you break that word down, it literally means process or in cell process of, so the process of going into the cell. And then exocytosis means out of cell process of, so movement of things into and outside of the cell. Okay, so the bulk level is going to require the use of those vesicles that we talked about when we were talking about the structure. Okay, these vesicles are going to fuse. So in both of these, we're going to say endo is into the cell, exo is out of the cell, and they're going to have to use the vesicles to fuse with the membranes and move the materials. All right, both of those types of movement require energy. Okay, so I guess I just want you to make sure you understand that. Okay, I don't think I have anything else for you, so thank you very much.